Alrighty, starting in 5, 4, 3, 2.
So my name is Ameur. I work at the SciLifeLab National Genomics infrastructure in Sweden. And we are a core facility for doing sequencing of all types. And today I'm going to talk about a project where we are using sequencing to look at CRISPR-Cas9 effects. So first, just a few words about the place where I work. So we are a core facility, and we are located both in the capital, in Stockholm, and also in Uppsala, which is another city. And this is a quite big operation. So we are 75 full-time employees, and we take on all different types of projects and have access to, I would say, most available sequencing technology. And during 2019, we processed about 1,000 projects and 34,000 different samples. For those of you who are not familiar with DNA sequencing, I just have a very brief and naive introduction to how this works. So basically what we do is to start from a sample. It could be a blood sample if we're working with human samples, for example, but we're also doing other types of organisms where we are starting from samples from other types of tissues, or it could be leaves or whatever. We then extract DNA, and in this picture I have it in form of nice long chromosomes. But what we then need to do is to fragment this DNA into small pieces. So these pieces are more than 100 base pairs long, and the reason for doing that is that the sequencing technology cannot read entire chromosomes, unfortunately. And with most technologies, we're fragmenting these into quite small pieces of about perhaps 200 base pairs. But what I'm going to talk about today is other technologies where we are looking at DNA molecules that could be several thousand base pairs and even maybe up to 100,000 base pairs in length. Once we have the DNA fragments, we put these onto our favorite DNA sequencer, and there are many different instruments for doing this type of next-generation sequencing. And finally, we end up with a pretty large data set that we need to analyze and interpret. And when it comes to this long-read sequencing technology that I will talk about today, there are different companies who is offering this service, and we have been using biosequencing for quite some time. So the first instrument that we installed was back in 2013, and to the left here you can see that it was a quite big piece of equipment and difficult to get into the lab. And more recently we installed a newer generation of the biosequencer to the right, which is a bit smaller and also generates a lot more data compared to this big instrument before. So a lot has happened in this technology. And when it comes to the technology itself, it produces DNA sequences for very long DNA fragments. And we can then start off with a DNA molecule that is perhaps 10 kbs in length. So this is to the top left. We then ligate adapter sequences to both ends of this DNA fragment. Then a polymerase is bound to one of these adapters. And when we have done that, we put this molecule onto a sequencing chip, and then the sequencing reaction starts. And to the right, you can see what happens during this sequencing reaction. So the polymerase will start doing sequencing, and it will go around this molecule many, many times, because as you can see, this is in fact a circular molecule. So we will create many copies of this strand. And what we then can do is to look at all these copies of the DNA fragment, and we can create a consensus read of this molecule. And this is something that can also be called a hi-fi read, and it has a very high accuracy. So we can get more than 99% accuracy of all the bases in this long DNA fragment, which is a quite unique feature of DNA sequencing. So this is one of the long-read technologies. We're also using another technology that is completely different, and this one is a nanopore-based technology from Oxford Nanopore. And the way this works is that we have a long DNA molecule that is fed through a pore, and when this molecule is being sort of pulled down through this pore, we get measurements of the currents when that happens. And in that way, we can sort of go through this trace and we can see which bases are being sequenced and then get readouts of the DNA molecules. And this is a quite interesting technology because it doesn't need any laser or anything to that to look at the sequence of the sequences. And this makes it possible also to create very small sequencers using this equipment. So in fact, there are portable instruments like this MinION to the bottom left, but then there are also higher throughput systems like the GridION and PromethION. And we have both the MinION and the PromethION system installed in our location. Okay, so what are we then doing with all these long reads that we create? And it can be used for many different purposes. We are taking on all types of projects that come to us from Swedish researchers and also from abroad. And it could be many different types of applications, both non-human or non-clinical, but then we also have a number of applications where we're trying to use this for more medical purposes. And one specific project that I can mention, because we are doing a lot of this non-human sequencing, and one of the biggest projects, I guess, that's happening at all now is the Earth Biogenome project, and we are taking part of this. And it's an effort to sequence basically all living organisms on the Earth. And long-read sequencing is actually quite a big part of this effort. But my specific focus, or sort of my research interesting, has been to see how we can use this type of technology for medical use and for human sequencing. And if you're interested in this topic, I was involved in writing this review article that was published a year ago, where we are looking at the advantages of using the long-read sequencing instead of the normal short-read sequencing. And I should mention that short reads are still sort of the norm when we're talking about NGS. But there are several advantages of using these long reads. And one thing that I would like to mention specifically is that these long reads allows us to look at things that we perhaps couldn't see before. So one thing is called here the concept of dark regions in the human genome. So as you know, I mean, humans have been sequenced for quite some time and are being analyzed with all types of technologies. But still there are some gaps in the human sequence if we're now using these short reads to look at the human genome. And this was a study from Genome Biology last year, where they looked at these regions where there are gaps. And this is one example where we have two genes in close proximity that are quite similar to each other. And if we look at these short reads, the Illumina reads at the top, you can see these gaps where we don't have any sequence information because it's difficult to align or impossible to align these reads uniquely to these regions. But if we look at this PacBio reads or the Oxford Nanopore reads, you can see that we cover this entire region. So these dark regions then are these gaps where we don't get any alignment in the short read data, but we can get good alignment with the long reads. Okay, so this was all just an introduction to the topic that I really want to talk about, and it's how we can use these technologies to study effects of CRISPR-Cas9 genome editing. I don't think I need to introduce Cas9 or the CRISPR-Cas9 system to you, because I mean it's well known that it's a revolutionary tool in life sciences. But one thing that has been discussed quite a lot is this concept about off-target effects. So it's unintended mutations that may be introduced when we are doing this type of genome editing. And there are different camps here. So there are some studies that show that there are a lot of these unwanted mutations that can occur when we're doing CRISPR-Cas9 editing. But then there have also been a number of studies that have shown that perhaps this is not really something that we need to worry too much about because we are not perhaps seeing them in living cells or in CRISPR-Cas9 edited organisms. And when we're looking at these off-target sites, the usual way to study them is to do a sequence comparison. And what we're doing then is basically to take the guide RNA sequence and we compare it to a reference genome to try to see where this guide RNA is binding or predicted to bind. And this is a quick and easy way to do this prediction, but there are also a few drawbacks here. One is that we need a reference sequence, otherwise we don't have anything to align it to. It might also be difficult to align these or to sort of predict where the guide RNA binds. I mean, is it really the same thing that happens in biology that happens in the computer? And also, the reference that we're using might be different compared to the cells that we're studying. There might be genomic variation. So can we do this in some other way? And there are, in fact, methods, sequencing-based methods to do this. For example, circle-seq, and yeah, there are a few other ones as well. But these methods are all limited by, first of all, that you're doing amplification and also that you're using short reads. This slide is just showing sort of the concept of genetic variation and guide RNA binding. So if we are now having a guide RNA, it might be designed to bind to this place here. So that's the on-target site if we're looking in the reference genome. But if we are now having an individual that has some variation compared to the reference, so it's indicated by these red arrows, maybe it's the case that this guide RNA will no longer bind at this on-target site, but instead it could bind at some other place in the genome. And one question we can ask ourselves here is if we can really study this off-target effect if we don't know the exact sequence of the individual that we are looking at. So we have been trying to look into this by using long-read sequencing, and we also have a preprint out on this project. And here I want to mention Ida Höjer, who is a PhD student who has been developing these methods that we're using here. And the methods that we have developed, we call them off-target sequencing, and there are two different protocols. So we have developed one protocol based on the PacBio sequencing, and this is called SMART off-target sequencing, or SMART OTS. And then we have the Oxford Nanopore version of this that is called Nano OTS. And basically, these protocols are quite similar. So we're starting off by looking at genomic DNA that has been fragmented, and then at some point we perform a Cas9 digestion of these molecules, we ligate some adapters, and then we perform sequencing. And one important thing to note here is that we don't do any type of PCR during this library preparation. So it's a completely amplification-free protocols. We applied these methods to the HEC293 cell line, and this is a human cell line, an embryonic kidney cell line. And what we also did with this cell line was to sequence this using long hi-fi reads, these high-quality reads from PacBio. And we sequenced this to 18x coverage. And in this way, we can get a pretty good idea of how this genome actually looks. And we can study all the bases in the genome and see where the guide RNAs bind and the exact sequences that they bind to. Okay, so the experimental setup we did here was to perform both these protocols, both the SMART sequencing and the nanopore sequencing, and we looked at 3 guide RNAs. So they're called ATXN10, MMP14, and NEC1. We also developed an analysis method to take the raw reads that we get from the sequencers to get good, accurate predictions of the guide RNA binding sites. And basically what we do is to align the reads to a reference. We then identify these Cas9 digestion sites, because what happens in the data is that the sites where the Cas9 digests, they will give rise to these types of peaks that are very sharp and where all the reads end at the exact same position where the Cas9 has cleaved them. And in the next step, we take all these peaks and we perform a pairwise alignment to identify which guide RNA that binds to each peak, and then we perform visualization of the output. This is one example of how it can look. So here we see the on-target site for these 3 guide RNAs. So we have ATXN10 to the left, MMP14 in the middle, and NEC1 to the right. And then we have the SMART sequencing and the nanopore sequencing data. The red line here corresponds to the Cas9 cleavage site. So you can see that we can clearly detect these sites in on-target sites with both these methods. But what was interesting is that we also get this type of signal also at other sites in the genome. So these are off-target sites. And here we have 3 examples of off-target sites for ATXN10, MMP14, and NEC1 that are found by both these methods. And for 2 of these guide RNAs, we have a 3 base pair mismatch when we compare the guide RNA sequence to the HEC293. And for the NEC1, we even have 5 mismatches, but still we get binding. So that's pretty interesting, I think, that we can see this type of binding, even though the guide RNA is quite different compared to the genomic DNA sequence. Here we see a visualization of the off-target sites that were detected by both these 2 methods. So these are like our high confidence off-target sites. So we have many of them for ATXN10. You can see at the top the guide RNA sequence, and then these colored boxes represents mismatches to this guide RNA sequence. And then we, in some cases, get signals even though we have insertion or deletion mismatches as well. One interesting observation is that we can also look at allele-specific Cas9 cleavage. So this is one example of a site for ATXN10, where we see that the guide RNA is preferentially binding to one allele. So at the bottom here to the left, you can see this hi-fi data. So this is the whole genome sequencing data. And here we have 2 different haplotypes. So we have the reference haplotype and we have the alternative haplotype. But if we look at the data that we get after our Cas9 enrichment, then we see only this alternative haplotype. And it turns out, if we look at the sequences, that this alternative haplotype has only 2 mismatches to the guide RNA, whereas the reference haplotype has 3 mismatches. So from this, we can conclude sort of that the genetic variation here, the SNP, can affect guide RNA binding, at least sort of in this in vitro experiment when we're looking at guide RNAs bound to DNA. We also were interested in seeing if we could find these guide RNA binding sites or these Cas9 cleavage sites without making use of the existing reference. So for this, we performed an assembly of all this hi-fi data. And this was done together with Jason Chin, who's an expert in this type of assembly work. And we were then able to get a good assembly of the HEC293 cell line. And then we could use this assembled version of the cell line to once again look at our data, map it to this de novo assembly, and see if we can find the cleavage sites. And it turns out that more than 98% of the cleavage sites could be detected in this way. So this means that we can perhaps start to use these approaches also for organisms where we don't have an existing reference. We also then looked into these dark regions that I mentioned before. So there was this paper from 2019 where they detected a lot of these dark regions that are also present in genes. And to the bottom here on this figure, you can see these red lines. So they correspond to these dark regions in one of the genes here called STRC. So what we did was to design guide RNAs in these specific regions. And you can see also from these Illumina whole genome sequencing reads. So this is from another project where we have whole genome sequencing data. We don't have any reads covering this position. But in our off-target sequencing assay, we get very nice signal here. So we can clearly detect off-target sequencing, or sort of, we can see that the Cas9 can cleave at this position. And that would not be possible to do with these approaches that are studying short read data. And this is just one other example of this. And in total, we had 6 of these guide RNAs that we designed in dark genomic regions, and we could recover the cleavage sites in all of them. So maybe this is a new way then to look at what happens in these like difficult regions of the genome when we're doing genome editing. Okay, but so far I've been talking about looking at guide RNAs or Cas9 cleavage just in genomic DNA. But of course, we're interested in what happens in living cells when we're doing genome editing. So to try to look at this, we performed this experiment on human fibroblast cells, where we first took then a bunch of fibroblast cells, and we performed this off-target sequencing using a specific guide RNA. And then we performed genome editing of these fibroblasts using the same guide RNA, and this was a vector-based approach for doing genome editing. And in the next step, we then took this edited fibroblasts, and we performed amplicon sequencing, long amplicon sequencing over all the predicted on and off target sites that were present here. So we amplified these regions and tried to see if there are any mutations occurring in the edited fibroblasts. And as control for this experiment, we also performed the same sequencing in the wild type fibroblasts. This slide shows some results. So this is genome editing using this MMP14 guide RNA. And at the top, you can see sequencing reads from this edited fibroblasts. And this flash here at the top, the red flash here, it corresponds to the position where the Cas9 cleavage occurs. And what we could see then is that we have a lot of small insertions and deletions at the Cas9 cleavage site. And this makes sense because the Cas9 is cleaved there, and then we have a DNA that has been repaired. But what was also interesting here is that we see some other things going on here, like this more than 500 base pair deletion in one of the molecules. And in some other place, we had a 328 base pair insertion here. And this insertion is in fact identical to the CRISPR-Cas9 vector. So we can see that the CRISPR-Cas9 vector jumped in at this location during the genome editing, which is kind of interesting. And if we're looking at the wild type fibroblast at the same position, we don't see much happening. So we see clear evidence of genome editing here. But what then happens at the off-target sites? So we performed the same experiment, and this is one of the off-target sites that I'm showing here. And in the edited fibroblasts and in the wild-type fibroblasts, we don't see any signal at all, which was kind of surprising. And we also quantified this. So this slide shows here in the middle, we have the Cas9 site, and to the left, we have the MMP14 on target. So you can see that we have an overrepresentation of insertions and deletions at the Cas9 site. But if we're looking at this off target, we basically don't see any signal. So from this, we can conclude that we don't see any off-target genome editing in this experiment that we performed. This is kind of surprising, I think, and it raises some questions. At least I had expected perhaps to see some off-target editing, but we don't see this in the fibroblast cells. So why could this happen? And there are many possible explanations. We don't know at this point. It could have something to do with the chromatin structure, perhaps. The chromatin is closed and cannot be edited. Maybe it's the CRISPR-Cas9 method we used. So we used this vector-based approach, which is kind of a gentle approach to do editing. Maybe some more rough method would have given more off-target mutations. Or could there be some kind of protective mechanism in the living cells that prevents this genome editing? Could it be something with the DNA repair in these fibroblasts, or something completely different? And we need to perform more experiments to figure this out. And one thing is to look at cancer cell lines and see if they behave in the same way. Okay, so coming back to the initial question, how worried should we be about these off-targets? I think this project has been a sort of like a bit of a roller coaster, because we were, in one way, we're kind of worried because we find a lot of off-targets in vitro when we're looking at guide RNAs bound to DNA. And we can see that the guide RNA can bind despite having many mismatches to the genome sequence. We also find these large structural variants at the on-target site, which could also be worrying if we want to perform this genome editing. But on the other side, we did not see evidence of off-target editing when we looked into living cells. So we cannot really say. I would say that we need to study this more to get a more clear picture of what's going on here. So the conclusions I have until now, I mean, what we have done here is to develop this off-target sequencing method. And using this approach and these hi-fi long reads, we are able to generate detailed map of Cas9 cleavage sites. We also can find evidence of allele-specific guide RNA binding, and we can detect off-target sites also, even if we're not making use of the existing reference. But I think that we still need to learn quite a lot about off-targets in living cells. And I have only been talking about human sequencing so far, but what about off-targets in other organisms? And I don't think it should be a big problem. I mean, it's relatively straightforward to do the sequencing and assembly of a genome to get a reference using this hi-fi sequencing, and then we are able to run this off-target assay. So it should be possible to predict off-targets also in other species. And with that, I would finally like to show which people have been involved in this project, and I would like to thank all of you for your attention.
Okay, great job. That was awesome. So pretty much from here, I'll just do some quick editing on the front and back end since there was no interruptions in between your presentation. And then I will have it ready for the event in a few weeks. Okay, great. So was nothing I need to sort of do again or anything? No, I thought it was great. Okay. You communicated clearly and it was understandable, so it was good.
Perfect.
Awesome. Thank you. Any questions for me before we wrap up here?
No, I don't think so. There's nothing else you need from me. You have all the material, I suppose. Will you also be like promoting this in some way before the event, or how's that working?
Yeah, we will be promoting as we do with our events, and then we'll also be sending you some promotional material as well if you were interested at all to promote on your end, whether it be social media or website. So you can expect to receive some banners and such from our team, most likely Lynn, and then you can promote as you like as well.
Okay, perfect.
Alrighty, and it looks like the only thing that I am missing from you are just a few learning objectives. So I can send a reminder as we get closer to the event, but just a few learning objectives for those continuing education credits for the audience.
Okay, yeah, sure.
Perfect. All right, well, if anything comes up, feel free to reach out, but if not, I will send you a reminder about those learning objectives as we get closer.
Perfect, thank you.
Have a good rest of your day. Bye-bye.
Yeah, bye-bye.